Americans spend $24 billion a year on fertilizer. Not commercial farms, but home gardeners. $24 billion to feed plants that for the previous 400 million years fed themselves just fine without anyone's help. There is a bucket sitting on a kitchen counter somewhere in rural Asia right now. Inside it are vegetable scraps, leftover food, and organic waste that would otherwise go into a trash bag. There is also a tablespoon of something that looks like dark powder. In 2 weeks, that bucket will produce more bioavailable plant nutrition than a bag of commercial fertilizer that costs $40. The person who owns that bucket did not buy anything to make it work. They used waste they were going to throw away anyway. This is not composting. Composting takes 6 months and cannot handle meat or dairy. This is something older, stranger, and significantly more powerful. And the reason you have never heard of it is simple. You cannot sell someone a bucket of kitchen scraps. So nobody tried. Let's talk about what synthetic fertilizer actually does to your soil, not what the label says. What it actually does. The pitch is straightforward: nitrogen, phosphorus, and potassium. 3 numbers on a bag. Pour it on your plants and they grow. It works. I am not going to pretend it does not work, because it does, in the short term, on the plant you are looking at right now. Here is what is happening underground while it works. Healthy soil is not dirt. It is a living system. 1 teaspoon of forest soil contains more microorganisms than there are people on Earth. These organisms, bacteria, fungi, protozoa, and nematodes, form a web of exchange with plant roots that has been running for 450 million years. Plants pump sugar down through their roots into the soil. Fungi and bacteria take that sugar and trade back minerals, water, and compounds the plant could not produce on its own. It is a barter economy. It works without any external input because the incentives are perfectly aligned. Synthetic fertilizer breaks that economy. When you pour soluble nitrogen directly onto the soil, the plant gets what it needs without trading anything. The fungi waiting at the root tips stop receiving sugar payments. They die back. The bacterial populations that fed on fungal byproducts shrink. The whole underground network, the one that took decades to build, collapses in a season or 2. And then the next season, you need more fertilizer, because the biological system that used to do the job for free is gone. A study from Wageningen University, published in 2021, looked at agricultural soils across Europe and found that microbial biomass had declined by 50 to 70% over the previous 60 years. 60 years is almost exactly the period when synthetic fertilizer use became standard. The researchers were not suggesting correlation, they were measuring collapse. This is not a niche concern. Degraded soil biology means lower yields over time, higher disease pressure, more water runoff, and less drought resistance. The fertilizer industry's solution to each of these problems is, predictably, another product. A fungicide here, a soil conditioner there, a small sachet of microbial inoculant at the garden center that contains organisms your soil used to grow on its own. The model is perfect if you sell inputs. It is ruinous if you are trying to grow food. I want to spend a minute on something that rarely gets talked about in gardening content. The Victory Garden program during World War II in 1943. The US government, facing food supply strain from the war effort, launched a national campaign to get civilian families to grow their own vegetables. At its peak, 20 million American families were tending victory gardens. Together, those gardens produced 40% of the nation's vegetables, not a small supplemental contribution. Nearly half the country's vegetable supply was grown in backyards and vacant lots by people with no agricultural training. The USDA manuals from that period are something to read. They are detailed, practical, and built entirely around biological fertility methods, compost, green manures, crop rotation, and animal waste. Not because the science of synthetic fertilizer did not exist yet, it did. But those materials were not available to civilians during wartime. The manuals taught people to close the loop, grow food, feed scraps back to the soil, grow more food. When the war ended, the factories that had been producing ammonia for explosives needed new markets. The chemistry for converting atmospheric nitrogen into ammonia, the Haber-Bosch process developed in Germany in 1909, was identical whether the end product was TNT or urea fertilizer. The transition took less than a decade. By the early 1950s, synthetic nitrogen fertilizers were being aggressively marketed to American farmers and gardeners as the modern scientific replacement for the old-fashioned biological methods the Victory Garden manuals had taught. The USDA shifted its extension programs. The universities restructured their agricultural curricula around synthetic chemistry. The Victory Garden knowledge, the composting techniques, the fermentation methods, the biological fertility systems did not get formally banned. They just stopped being taught. And within a generation, most of it was gone from mainstream practice. What I find hard to shake is that the timing is not ambiguous. The knowledge was there. The results were documented, and the decision to stop teaching it was not made because it stopped working. It was made because it stopped being profitable. Now let me introduce you to two people the fertilizer industry would prefer you had never heard of. The first is Cho Han Ki-yu, Korean farmer in the 1960s. Not a scientist, not a professor. A man who couldn't afford to buy inputs and decided to find another way. He spent years observing the forest soil near his farm. The same observation Fukuoka made in Japan around the same time. Untouched forest floor is extraordinarily fertile. Nothing added to it, nothing removed. Just continuous cycles of organic matter falling, decomposing, and feeding the next generation of plants. He asked a question that sounds obvious once you hear it, but apparently nobody in the agricultural establishment was asking. What if you could capture the microorganisms doing that work and multiply them deliberately? He spent years developing what became Korean natural farming, a set of fermentation-based preparations made entirely from local, free, or nearly free materials. The goal was not to replace nutrients. It was to restore the biological engine that produces and cycles nutrients on its own. The University of Hawaii tested his system in the early 2000s. Results on test crops were comparable to conventional fertilizer programs, with the additional finding that soil quality measurably improved over time instead of degrading. Approximately 14 cents per square meter. The second person is Dr. Teruo Higa, an agronomist at the University of the Ryukyus in Okinawa, Japan. In the early 1980s, he was working with various microorganism cultures when he made an accidental discovery, one he spent the next decade trying to explain. He had been disposing of spent cultures by dumping them on a patch of unused ground near his laboratory. After a few months, that ground was noticeably different from everything around it. More plants were growing, greener and more vigorous growth. Earthworms appeared in soil that had shown no earthworm activity before. He analyzed what he had been dumping. It was a mix of lactic acid bacteria, yeasts, and phototrophic bacteria, three groups that do not normally coexist in high concentrations in nature. Together, they were doing something that neither group did alone. He spent years working out why. Lactic acid bacteria produce compounds that suppress pathogens. Yeasts produce hormones and enzymes that stimulate root growth. Phototrophic bacteria synthesize amino acids and bioactive compounds using light energy. Each group also produced metabolites that the other groups could use as food. They were not just coexisting, they were feeding each other, creating a self-sustaining consortium more stable and more active than any single species. He called them effective microorganisms, EM for short. A meta-analysis published in Applied Soil Ecology in 2019 reviewed 40 independent studies on EM applications across different crops and climates. The average increase in soil microbial biomass was 35%. Reduction in soil-borne pathogens was measurable in 78% of cases tested. Yield improvements averaged around 15% over control plots, with the more significant finding that improvements compounded over successive seasons rather than declining. That last part is worth sitting with. Most fertilizer interventions produce diminishing returns. EM-treated plots showed increasing returns. The biology was rebuilding itself. Higa published his findings, and he started a company to distribute EM cultures. Then something predictable happened. The agricultural establishment largely ignored it. A few university extension programs tested it, and some organic farming circles adopted it. But it never entered mainstream agricultural education in the West because there was no mechanism to profit from soil biology that farmers could capture and replicate themselves for free. Which brings us to bokashi. Bokashi means fermented organic matter in Japanese. It is the most accessible application of Higa's research. It uses the EM consortium to ferment kitchen waste anaerobically, without oxygen, the way kimchi or sourdough ferments, producing a pre-digested soil amendment that the microbial ecosystem can process in 2 weeks, instead of 6 to 12 months for a traditional compost pile. The key difference from composting is what it can handle. Regular compost cannot take meat, fish, dairy, or cooked food without attracting pests and producing pathogens. Bokashi takes all of it. The anaerobic fermentation process acidifies the material quickly enough that pathogenic bacteria cannot establish. The result smells sour, vinegary, and pickled, not rotten. The liquid that drains from the fermentation vessel during the process is particularly concentrated. Diluted with water and applied to soil or sprayed on leaves, it introduces active microbial colonies, organic acids, enzymes, and growth compounds directly into the plant's environment. Changes in plant vigor appear within 48 hours of application. Commercial liquid biofertilizer at a garden center costs between $8 and $15 per liter. Bokashi liquid is a passive byproduct of a process that runs on waste. The Rodale Institute published a comparison study in 2020 looking at microbial compost teas versus synthetic fertilizers on vegetable crops grown in degraded soil. Yield results were equivalent in year 1. By year 2, the compost tea plots were outperforming the synthetic plots. By year 3, soil organic matter had increased by an average of 0.4%, which does not sound significant until you realize that rebuilding 1% of soil organic matter through conventional means takes roughly a decade. What makes bokashi work is not any single ingredient. It's the consortium. The lactic acid bacteria acidify the environment fast enough to suppress mold and pathogens. The yeasts break down complex carbohydrates into simpler compounds the bacteria can use. The phototrophic bacteria process light and chemical energy into amino acids and bioactive substances. Each organism feeds the others. The system is self-reinforcing in a way that no single species product can replicate, which is precisely why it can't be cleanly bottled and sold at a markup. When the fermented material is buried in soil, the pH difference between the acidic bokashi and neutral soil triggers a rapid cascade of microbial activity. The organisms that rush in to neutralize and process the material are doing exactly what they evolved to do, breaking down complex organic material into mineral compounds plant roots can absorb. The fermentation hasn't replaced soil biology. It's given soil biology the most bioavailable food source it's ever encountered, in a form that bypasses months of slow surface decomposition. The researchers who find this genuinely interesting tend to be in soil science departments. The ones who find it inconvenient tend to be in agronomy programs funded by fertilizer companies. That is not a conspiracy. It's just how research funding shapes research questions. Fermented organic fertilizers are not a modern invention. They are not even a 20th century invention. Across Asia, fermented plant and animal waste has been used as a soil amendment for thousands of years. Agricultural records from ancient China describe fermentation methods for organic waste that produce amendments strikingly similar in principle to what Higa and Cho were formalizing in the 20th century. In Korea, the tradition of fermenting food scraps for agricultural use predates written records. In Japan, fermented organic fertilizers were used in rice paddy cultivation for centuries before synthetic alternatives existed. These were not primitive approximations of something better. They were refined systems that worked well enough to feed dense populations on relatively small amounts of land. The colonial period and the Green Revolution largely erased these practices from mainstream agriculture. Not immediately and not uniformly, but the combination of government promotion, subsidized pricing, and educational restructuring pushed synthetic inputs to the center of global agriculture within about 30 years. What Cho and Higa did was look at what had been lost and build a scientific framework around it. They were not inventing new biology. They were explaining why something that had worked for centuries actually worked. In 2024, a small number of multinational companies controlled the majority of the global commercial fertilizer market. Combined agrochemical revenues were over $200 billion a year. A farming method that converts kitchen waste into fertility, using organisms from the local soil environment, does not generate $1 of revenue for that system, so it does not get shelf space. It does not get university extension programs. It does not get recommended by the staff at garden centers trained on commercial product lines. The mechanism of erasure is subtle. When Higa's EM research started gaining traction in the 1990s, the response from the agricultural establishment was not to argue that it did not work. The response was to demand large-scale, multi-year, multi-site randomized controlled trials, the same standard applied to pharmaceutical drugs. Independent researchers with no industry funding cannot run those trials, so the research stays in smaller journals. Extension services do not adopt it, garden centers do not stock it, and the average gardener never hears about it. This is how most inconvenient agricultural knowledge gets handled. Not banned, just unfunded, unextended, and untaught until the generation that knew it dies off. Meanwhile, tens of thousands of farmers in Japan and South Korea use Korean natural farming and bokashi as their primary fertility program. Young-sang Cho, the son of the system's founder, has published all Jada methods as open source, free to anyone, specifically to prevent commercialization. In Brazil, Mexico, and India, farmer networks run bokashi programs at scale. The technique is being used in urban agriculture projects across Southeast Asia. None of this appears in mainstream American agricultural media. You can't patent kitchen scraps, and that's the problem. Bokashi won't fix completely depleted soil overnight. Starting with heavily compacted, chemically saturated ground, you are looking at 2 to 3 seasons before the biological system really establishes. It does not deliver a predictable number on a soil test the week after application. It is a process. The results build over time, and crucially, they keep building, which is the opposite of what happens with synthetic inputs. There is also something worth acknowledging about scale. Bokashi works for a household garden. It works for a small farm. Whether it scales to feed industrial agricultural systems at the volumes they currently require is a genuinely open question. The Green Revolution did increase yields dramatically. The cost was the biological degradation of the soil that produced those yields. Whether we can have both productivity and biology is the central question of 21st century agriculture, and the honest answer is that nobody fully knows yet. What bokashi demonstrates, clearly and repeatedly across 40 years of independent research, is that the relationship between soil biology and plant fertility is not a historical curiosity. It is an active, functioning system that modern agriculture has been systematically undermining. The tools to rebuild it are not expensive, not technical, and not dependent on any supply chain. What it does is closes a loop that the fertilizer industry spent 50 years convincing us needed to stay open. You eat food, that food produces waste. That waste, processed correctly through anaerobic fermentation, feeds your soil. The soil feeds your next crop. There is no step in that cycle that requires a purchase. Cho Han-kyu understood that in the 1960s in rural Korea because he had no choice but to find another way. Teruo Higa documented the science behind it in the 1980s in Okinawa because a patch of ground near his lab kept getting more fertile instead of less. The research has been accumulating ever since in journals that the fertilizer industry has no financial interest in amplifying. The most expensive thing in your garden is not the fertilizer. It is the habit of buying it. Everything you need is already being thrown away in your kitchen every day.